In our first SoftChalk lesson of Unit 1, Lecture 5, we're going to be looking at the Bacterial Glycocalyx, bacterial capsules, and biofilms. Let's start out again by looking at our fundamental statements or bullet points for this SoftChalk lesson. All bacteria secrete some sort of glycocalyx, which is an outer viscous covering of fibers extending from the bacterium. An extensive, tightly bound glycocalyx adhering to the cell wall is called a capsule. Phagocytosis involves several distinct steps, including the attachment of microbes to the phagocyte through unenhanced or enhanced attachment, followed by ingestion of the microbe, its placement into a phagosome, and the destruction of microbes after fusion of the lysosomes with the phagosome. Capsules enable bacteria to resist unenhanced attachment by covering up bacterial pamps so they're unable to bind to endocytic pattern recognition receptors. The glycocalyx also enables some bacteria to adhere to environmental surfaces, colonize, and resist flushing. The body's adaptive immune defenses can eventually overcome bacterial capsules by producing opsonizing antibodies, primarily IgG, against the capsule that are able to stick the capsule to the phagocyte. Biofilms are groups of bacteria attached to a surface and enclosed in a common secreted adhesive matrix and are functional, interacting, and growing bacterial communities. Most bacteria in nature exist as biofilm populations. Many chronic and difficult to treat infections are caused by bacteria in biofilms. An antigen is a molecular shape that reacts with antigen receptors on lymphocytes to initiate an adaptive immune response. The actual portion or fragment of an antigen that reacts with antibodies and with receptors on B lymphocytes and T lymphocytes are called epitopes. Adaptive immunity refers to an antigen-specific defense mechanism that takes several days to become protective and is designed to react with and remove a specific antigen. This is an immunity we develop throughout life. Humoral immunity involves the production of antibody molecules in response to an antigen. Cell-mediated immunity involves the production of cytotoxic T lymphocytes, activated macrophages, activated NK cells, and cytokines in response to an antigen. These defense cells help to remove infected cells and cancer cells displaying foreign epitopes. And finally, the bacterial glycocalyx or capsule functions as an antigen and can trigger the production of opsonizing antibodies that bind to epitopes on the glycocalyx. These opsonizing antibodies can bind bacteria to phagocytes for more efficient phagocytosis. So those are our bullet points, and let's look at our detailed learning objectives for this SoftChalk lesson. You should be able to state the chemical composition and two common functions of a bacterial glycocalyx. You should be able to briefly describe the following steps in phagocytosis: unenhanced attachment, enhanced attachment, engulfment, and destruction. Briefly describe how a capsule might initially enable some bacteria to resist being phagocytosed by white blood cells. Briefly describe how opsonizing antibodies made against bacterial capsules help to protect the body. Define biofilm and state at least 3 advantages of biofilm formation in bacteria. Compare and contrast innate immunity and adaptive immunity. Define antigen and epitope. Define humoral immunity and cell-mediated immunity. And briefly describe how opsonizing antibodies can promote phagocytosis. And we have one highlighted bacterium in this SoftChalk lesson, Streptococcus pneumoniae. So let's start out by looking at the bacterial glycocalyx. Now, all bacteria secrete some sort of glycocalyx, And a glycocalyx is an outer viscous covering of fibers that extend from the bacterium. If we look at the electron micrograph in figure 1, we see these fibers here coming off of the cell wall, and that represents the glycocalyx of that bacterium. Now, this is a very thin glycocalyx. If we saw this bacterium under a regular microscope, where it was about the size of one of these bacteria in figure 2, we wouldn't even see that glycocalyx. So although all bacteria have a glycocalyx, in some cases they're very thin coverings. If the glycocalyx appears as an extensive, tightly bound accumulation of gelatinous material adhering to the cell, where the cell is embedded in this gelatinous material, then the glycocalyx is often called a capsule. And in figure 2, we see a capsule stain of Klebsiella aerogenes that we'll be looking at in lab 6. So we see the tiny bacilli in the center, and the clear covering around them is the bacterial capsule, an extensive glycocalyx. In terms of structure and composition, the glycocalyx is usually a viscous polysaccharide or polypeptide slime. And whether or not the bacterium is able to produce a glycocalyx does depend often on environmental conditions. Figure 3 shows a capsule stain of normal flora of milk called Streptococcus lactis, where you can easily see a chain of streptococci surrounded by this clear capsule or glycocalyx. And there's a little self-check on the structure of bacterial glycocalyces. Now let's look at the function of bacterial glycocalyces and capsules and their significance to bacterial pathogenicity, the ability to cause disease. There's many functions that have been associated with the glycocalyx, but there's two very important ones we're concerned with here in our course. First of all, the glycocalyx enables certain bacteria to resist phagocytic engulfment by white blood cells in the body, or by protozoan in soil and water. In a second function, the glycocalyx enables some bacteria to adhere to environmental surfaces, such as rocks or our teeth or body cells, in order to colonize and to resist flushing. Now, before we get into how the glycocalyx serves as a mechanism in bacterial pathogenicity, we need to understand the steps in phagocytosis. You did cover these in biology when you had your prerequisite course for microbiology, and we will cover this in detail in Unit 5, but we need to understand the steps now to see how bacteria can resist phagocytosis. So we're going to go through the steps of phagocytosis. Now, the first step in phagocytosis is called attachment. And this is where the surface of the microbe has to attach to the cytoplasmic membrane of the phagocyte. Attachment of microorganisms is necessary for the phagocyte to pull the bacterium in or ingest it. And attachment can either be unenhanced or enhanced. And we'll be talking about that quite a bit during the semester. So let's start with unenhanced attachment of a microbe to a phagocyte. Now, this involves recognition of our old friends, a pathogen-associated molecular pattern, or PAMPs, found on the surface of bacteria. Components of common molecules like peptidoglycan, teichoic acids, lipoteichoic acids, mannans, and such in the cell wall, and not found on human cells, are PAMPs that we learned about previously. And these PAMPs can attach to what are called endocytic pattern recognition receptors on the surface of phagocytes. So the pattern recognition receptors, or PRRs, that we talked about previously when we looked at the gram-positive, gram-negative cell wall and such, those were pattern recognition receptor where, after they react with PAMPs on bacteria, triggered inflammation by causing the synthesis and secretion of inflammatory cytokines. But there's another category of pattern recognition receptors that are found on the surface of phagocytes, and they're called endocytic pattern recognition receptors. So endocytosis, remember, is a combination of phagocytosis and pinocytosis. When something is engulfed and placed in a vesicle, that's called endocytosis. And so these are pattern recognition receptors that promote endocytosis. So an unenhanced attachment, this is where PAMPs on the surface of the microbe, such as cell wall PAMPs and other PAMPs, bind to endocytic pattern recognition receptors found on the surface of phagocytes. And this is the first step attachment that we see during phagocytosis, because innate immunity involving PAMPs and PRRs is our first line body defense. That becomes activated within hours after exposure to a microbe. And we are born with endocytic pattern recognition receptors that fit all the common PAMPs on microorganisms for the purpose of unenhanced attachment. And we can see that nicely in our first animation here. So here we see some PAMPs on the surface of a bacterium. Again, that could be some cell wall components like peptidoglycan or LPS or teichoic acids or whatever. And we have endocytic pattern recognition receptors on the surface of phagocytes, receptors that promote endocytosis, that is phagocytosis and pinocytosis. So in this case, the endocytic pattern recognition receptors fit the PAMPs. As pseudopods go out, the endocytic pattern recognition receptor binds to the PAMP, retracts, and attaches the bacterium to the cytoplasmic membrane of the phagocyte. And again, the organism has to be attached to the phagocyte before it can be pulled in. Now, enhanced attachment, our other form of attachment, is the attachment of microbes to phagocytes by molecules. Primarily, this is by antibody molecules, specifically those called IgG, or by proteins produced during the complement pathways, another part of innate immunity. And the attachment structures on the surface from the complement pathway are called C3b and C4b. So what we're saying here is that molecules we make during body defense, like IgG produced during humoral immunity or antibody production, and C3b produced by the complement pathway, can stick microbes to phagocytes, act like a molecular cement that attaches the bacterium to the phagocyte. And so these molecules that promote attachment for enhanced attachment are called opsonins. And these opsonins then play a role in what we call opsonization. And that's where molecules called opsonins like IgG or complement proteins like C3b attach microbes to phagocytes. And enhanced attachment is much more specific and efficient than unenhanced attachment. But that might take longer to develop because this can be part of adaptive immunity in the case of antibody molecules, which can take several days to become protective. So here's an animation that illustrates enhanced attachment or opsonization by the most common antibody we make called immunoglobulin G. So during adaptive immunity, we recognize epitopes on the surface of antigens of bacteria, and we make antibodies where the FAB or antigen binding fragment of the protein antibody molecule we make is tailor-made to fit these epitopes on the surface of the bacterium. So as we see here, the FAB or antigen binding part of the antibody fits the epitopes and locks on. And then the bottom part of the antibody, called the FC portion, can bind to FC receptors on the surface of phagocytes. So again, as pseudopods go out, the FC receptor can bind to the FC portion of IgG, and that antibody kind of superglues the bacterium to the surface of the phagocyte so it's firmly attached and can be pulled in and eventually ingested. Following attachment by unenhanced or enhanced attachment, The next step of phagocytosis is called engulfment. And this involves the polymerization and depolymerization of part of the cytoskeleton of our phagocytes called actin filaments. And as these actin filaments polymerize, they push the membrane of the phagocyte up and around where they eventually fuse, and the pseudopods then engulf the microbe and place it in a vesicle called a phagosome. So remember, the phagosome is the vesicle that forms during phagocytosis, when the organism is engulfed and placed in a membrane-bound sac called a phagosome. And we see this occurring in the illustration here in figure 7. Once that pinches off and is pulled in, then we see that the organism in figure 7 here is placed inside a phagosome. So that's the engulfment, pulling the organism in and placing it in a membrane-bound sac called a phagosome. And so here we see that much happening with unenhanced attachment, where PAMPs are going to bind to endocytic pattern recognition receptors. So again, the endocytic pattern recognition receptor binds to an appropriately shaped PAMP. As actin polymerizes, pseudopods are formed, they engulf the organism and place it in a phagosome. And then in the next step, lysosomes, the membrane-bound sacs that contain the killing chemicals that are going to lyse the bacteria, move along the cytoskeleton and eventually fuse with the phagosome, forming what's called a phagolysosome. And at that point, then, the digestive chemicals and the toxic oxygen molecules can kill the microbe and digest it. And here's a summary of phagocytosis with enhanced attachment, where again, in this case now, we're making antibodies, IgG, that fit epitopes on the bacterium, epitopes of antigens on the bacterium. So the FAB part of the antibody, again, is going to, there we go, get the, so here we see the FAB part of the antibody binding to the epitopes on the bacterium, and then the FC part of the antibody, or bottom part that directs biological activity, can bind to FC receptors on the surface of the phagocyte, binding it tightly to the surface. And again, through polymerization and depolymerization of actin, the organism is engulfed and placed in a phagosome. And then the lysosome, the membrane sac containing the digestive chemicals and killing chemicals, will travel along the cytoskeleton, fuse with a phagosome to produce a phagolysosome. And at that point, the organisms are killed and digested. So here's a nice little movie showing a human neutrophil, the most abundant white blood cell we have, phagocytosing some methicillin-resistant Staph aureus. So here's a neutrophil. These are MRSA. And you see it sends out pseudopods. It can engulf whole groups of bacteria here. And as they engulf them and pinch off, they place them in the membrane-bound sac called a phagosome. And then eventually lysosomes are going to fuse with the phagosome and kill and digest those organisms. Here we see one at the bottom also doing that. And here's the second movie showing some yeast being phagocytosed. So these are yeast cells. This is a neutrophil. And we see the neutrophil crawling towards the organism through chemotaxis, actually. It starts sending out pseudopods here, and they engulf the yeast and place it in a membrane-bound sac called a phagosome. And here we see another one being engulfed by the neutrophil down here. And once they're engulfed and placed in a phagosome, they can be digested by lysosomes. So once they're engulfed, the final step is the destruction. And that's again where lysosomes, these are organelles that contain digestive enzymes, often called acid hydrolyses because they're designed to work at an acid pH that we'll talk about later. And they can digest the engulfed microorganisms. So the lysosomes contain the digestive enzymes and other microbicidal chemicals. As the lysosome, the sac containing the killing chemicals, fuses with the phagosome, the sac containing the bacteria, the microbes are destroyed. And during the process with the two fuse, that's called a phagolysosome, shown there in figure 8. And there's a little self-check you can do on that. So anyway, those are the steps in phagocytosis, and we're going to start learning them now. We'll go through these steps again in Unit 3 when we talk about how bacteria can resist phagocytosis. And then in Unit 5, when we get to innate immunity, we'll have this in detail, the steps in phagocytosis. So if we learn these steps now, we'll know them by the time we get to Unit 5. So getting back to our topic here, the function of the glycocalyx, now we're going to see how that glycocalyx plays a role in resisting phagocytosis. And we're going to be using glycocalyx and capsule kind of interchangeably here, since often that glycocalyx is a rather massive mass called a capsule. So what capsules do is they resist unenhanced attachment. They prevent the endocytic pattern recognition receptors on the phagocytes from recognizing the pathogen-associated molecular patterns or PAMPs found on the bacterial surface. So the bacterium's not stuck to the phagocyte. And so what capsules do primarily is they resist unenhanced attachment, our first line attempt to engulf the bacteria. And that means since we're not removing the bacteria initially through unenhanced attachment, the bacteria can continue to replicate and possibly cause harm before we can eventually stick the encapsulated bacteria to phagocytes by enhanced attachment with the antibody IgG. So again, here we see in figure 10 that this capsule is rather extensive and it's covering up the PAMPs on the surface of the bacterium. And by covering up the PAMPs are unable to bind to the endocytic pattern recognition receptors on the surface of the phagocyte. And we see that happening in this next animation here. So there's the capsule surrounding the bacterium and covering up the pathogen-associated molecular patterns or PAMPs on the surface of the bacteria, and that's going to prevent them from binding to the endocytic pattern recognition receptors on the phagocytes during unenhanced attachment. So since the endocytic pattern recognition receptors can't bind to the PAMP, the bacterium is never attached to the surface, it's never engulfed, it's never destroyed. And that's the primary role of capsules in resisting phagocytosis. They block unenhanced attachment by covering up PAMPs, so the PAMPs are unable to bind to endocytic pattern recognition receptors involved in unenhanced attachment. And there's a lot of bacteria that produce capsules to resist phagocytosis, including the pneumococcus, streptococcus pneumoniae, Haemophilus influenzae type B, Neisseria meningitis, Bacillus anthracis, and the whooping cough bacterium Bordetella pertussis, to name a few. But the body has a way around that capsule. If we couldn't ingest this organism, it would continue to replicate and could possibly cause disease. So we use adaptive immunity to get rid of that capsule. And we do this by producing opsonizing antibodies, the antibody IgG, against epitopes of the capsule. So we recognize the sugars branching out of the polysaccharide, the epitopes of a polysaccharide as foreign, and we eventually make antibodies that fit the epitopes of that capsule. But again, that can take up to 5 to 10 days if we've never seen this bacterium before we make protective levels of antibodies. And during that 5 to 10 days, it's resisting unenhanced attachment. But that is eventually our defense against that. So here we see opsonizing antibodies removing encapsulated bacteria. So the body recognizes epitopes of the bacterial capsule as foreign and eventually makes antibody molecules that fit those epitopes. The FAB, or antigen binding portion of the antibody, the tips of the antibody, bind to the epitopes of the capsule. And then the FC, or bottom part of the antibody, binds to FC receptors on phagocytes. And in this way, we can eventually stick encapsulated bacteria to the surface of the phagocyte, where they can then be engulfed, put in a phagosome, and eventually destroyed with lysosomes. And here we have our highlighted bacterium, the Pneumococcus streptococcus pneumoniae, as an example of an encapsulated bacterium that resists phagocytosis with a capsule. Now, the other important function of the glycocalyx is that it can allow bacteria to adhere to and colonize environmental surfaces. Bacteria found growing in the river under rocks often have biofilms of capsules there that allow them to adhere. The bacteria that form the microbiota of your teeth, or plaque on your teeth, form a biofilm there. So the important thing here is that the glycocalyx does enable some pathogens to colonize cells or surfaces of our body and thus resist flushing. Virtually every part of our body has a flushing action, but again, if the bacteria can adhere, they can resist flushing. Now remember, we did see back earlier under cell walls that most bacteria produce cell wall adhesins that allow them to adhere to receptors on host cell and colonize and resist flushing. This is a second way bacteria can adhere, by means of a glycocalyx, and eventually, as we'll see in a few minutes, producing a biofilm. And later at the end of this unit, we'll see a third way bacteria can adhere using special surface structures called pili. And often the body's microbiota, or normal flora, do produce capsular polysaccharides, or glycocalyx, that eventually lead to biofilm formation on host tissue, which will be our next topic in this SoftChalk lesson. So those are the two roles of the capsular glycocalyx that we're concerned about: the ability to resist unenhanced attachment by covering up PAMPs so they can't bind to endocytic pattern recognition receptors. But our way around that is we can eventually stick capsules to phagocytes through opsonization by using opsonizing antibodies. And the second role is involved in adhering to surfaces such as body cells, and that allows bacteria again to colonize and resist flushing. So let's look at the significance of the glycocalyx in initiating body defense. Now, we talk about this in a number of SoftChalk lessons in Unit 1, so that by the time we get to Unit 5 on innate immunity and Unit 6 on adaptive immunity, we're familiar with a lot of definition that we'll need at that time, which will make it much easier at the end of the course when we get to Units 5 and 6. So in terms of initiating adaptive immunity, the polysaccharides, or proteins that form the bacterial capsule, function as antigens, and they initiate adaptive immunity against the antigen of that capsule. So remember, an antigen is a molecular shape that binds to antibody molecules once we make them, but also binds to antigen receptors on lymphocytes to initiate adaptive immunity. We recognize antigens as foreign because epitopes of those antigens will fit receptors on B and T lymphocytes, telling us that's a foreign antigen rather than one of our own antigens, or self. And again, the actual portion or fragment of the antigen that actually binds to the antibody or binds to the receptors on B and T lymphocytes are called epitopes. And again, as we've said several times, an epitope might be a group of maybe 5 to 15 amino acids with unique shape sticking out of a protein antigen, like we see in figure 11a, where along this long protein molecule, there'll be a few amino acids maybe here sticking out that have this shape. there'll be a different order of amino acids here sticking out that have this shape, and another group of amino acids here sticking out that have that shape. So a protein antigen often has many different epitopes because there's 20 building blocks in proteins, giving us a large variety of different arrangements of amino acids, giving us a wide variety of different shapes to recognize. Now, if that is a polysaccharide antigen, such as most bacterial capsules, then the epitope would be 3 to 4 sugar residues branching off of a polysaccharide antigen. So as we see in figure 11b, a polysaccharide may be many sugars long, but typically there's only 1 or 2 different sugars that make up that polysaccharide with very limited shapes. So the epitopes are actually a few sugars branching off of the polysaccharide. The polysaccharide being an antigen, the sugars branching off being an epitope. Just like in figure 11a, the protein is an antigen, but these little shapes sticking out, different orders of amino acids, are epitopes. And a single microorganism often has many hundreds of different shaped epitopes that our lymphocytes can recognize as foreign. So it's not like we can make one antibody that fits a bacterium. We make an antibody that fits every epitope of every protein and every polysaccharide associated with that organism, allowing us to mount a very vigorous adaptive immune response against that antigen. So again, the body recognizes an antigen as foreign when epitopes of that antigen bind to receptors on B lymphocytes and T lymphocytes, the two groups of white blood cells that carry out adaptive immunity. And there are 2 branches of adaptive immunity. Humoral immunity, again, is the production of antibody molecules in response to an antigen, and that's carried out by B lymphocytes. And once we make these antibodies, these can remove or neutralize microorganisms or neutralize toxins by binding to epitopes. And the one we talked about here, of course, was opsonization. We showed you how that works a minute ago. So in this case, the epitopes would be sugars coming off of the capsular polysaccharide of this bacterium. And again, we make antibodies that are going to eventually fit epitopes of that capsule. The FAB part of the antibody binds to the epitope of the capsule, and then the other end of the antibody binds to FC receptors on phagocytes for enhanced attachment or opsonization. And thus the antibody cements the capsule to the phagocyte so it can be engulfed and eventually destroyed with lysosomes. And then cell-mediated immunity is a cellular response where we produce cytotoxic T lymphocytes, activated macrophages, activated NK cells, and cytokines in response to an antigen. And that's carried out by T lymphocytes. And we use this more to remove infected cells and cancer cells that are displaying foreign epitopes. So again, that's just a review of some definitions we've had several times that we'll be taking up in more detail these terms in Unit 6 in adaptive immunity. So if we learn them now, that'll make it much easier later in the semester when we kind of run near the end of the course. And there's a little self-check you can do on that. Now, often the glycocalyx production eventually leads to bacteria producing what are called biofilms. So many pathogenic bacteria, as well as many of our normal flora or microbiota, form complex communities in the form of biofilms. So we define a biofilm as a group of bacteria attached to a surface by means of the glycocalyx and enclosed in a common secreted adhesive matrix, typically polysaccharide. In other words, that is the glycocalyx or capsule. So as the bacteria start replicating and secreting the glycocalyx, you get a number of bacteria enclosed in a common glycocalyx. And again, we're calling it a secreted adhesive matrix because it's allowing the bacteria to adhere to cells. Now, we're going to learn later on in Unit 2 when we talk about quorum sensing that bacteria and biofilms do often communicate or talk to one another chemically through a process called quorum sensing. and they're able to interact with and adapt to their environment as a population of bacteria rather than as individual bacteria. So we're going to see later in Unit 2 that bacteria initially gain a foothold and colonize the body by functioning as individual bacteria, every bacterium acting for itself, trying to be the one that's able to adhere to cells and colonize. But eventually, when a certain density of bacteria accumulates, that turns on virulence genes that allow the bacteria to function as a population of bacteria, where they're all benefiting from being in the population rather than as individual bacteria, each out for itself. So by living as a community of bacteria in a biofilm, a population, this gives several advantages to bacteria. First of all, they can better resist attack by antibiotics. When you have a large group of bacteria embedded in this common polysaccharide slime, it's harder for the antibiotics to diffuse through and get to the bacteria. Also, the bacteria in the biofilm tend to be replicating more slowly, so it would take longer for the antibiotics to be effective. Secondly, that biofilm traps nutrients for bacterial growth, so they remain in a favorable niche. They eventually start producing enzymes and toxins that can damage the cells they're adhering to, so nutrients leak out and start feeding the biofilm. And of course, as we just saw, the biofilm can adhere to environmental surfaces such as cells in the body or the teeth in the case of plaque and resist flushing. The bacteria live in close association with one another in the biofilm and are communicating with other bacteria in the biofilm through quorum sensing. And by being in a biofilm, they're better able to resist phagocytosis and attack by the body's complement pathways. So biofilms are functional, interacting, and growing bacterial communities. And they begin to mature with time. The biofilms eventually contain their own water channels that deliver water and nutrients throughout the biofilm community. So they can be very complex little neighborhoods. For example, this illustration here shows a photomicrograph of a biofilm of Pseudomonas. And so here we see the Pseudomonas entrapped in this common adhesive matrix or glycocalyx, which is forming now a biofilm, but eventually water channels form in that biofilm. And those water channels then bring in water and nutrients and remove waste products. They can bring in oxygen if they need oxygen. And as those water channels run throughout the biofilm, the whole neighborhood winds up getting the water and the nutrients they need. We see some other nice pictures of biofilms in this link here, showing a biofilm of Haemophilus influenzae. Now, this is a bacterium that causes a lot of respiratory infections. And for Haemophilus influenzae type B, the capsule is a major virulence factor. By the way, this does not cause influenza. It was named long before we really knew about viruses, and they thought at one time maybe this caused influenza, but it was just a secondary bacterial invader, but the name still holds. So if we scroll down here, we'll see some nice electron micrographs of biofilms. So Haemophilus influenzae is a bacillus-shaped bacterium, but here you see it's all encased in this common adhesive matrix, this glycocalyx, the slimy glycocalyx. And as the bacteria begin to grow and spread out, they start developing water channels that start running through the population in order to deliver nutrients and oxygen and water and other such things. Here we see the water channels further developed with the bacillus-shaped bacteria still in the glycocalyx. And especially in this illustration here, we see it nicely. So this is a major water channel, one of these up here. But then if you picture this like a neighborhood, this would be a major road going to the neighborhood, and these would be the minor roads running throughout the neighborhood. These are minor water channels so that all parts of that biofilm are eventually getting nutrients and oxygen, water and such. Now we're going to learn more about the biofilm formation when we get to quorum sensing in Unit 2. And these couple paragraphs here talk about Pseudomonas aeruginosa, which we'll use as an example of a bacterium with quorum sensing later on. We're going to skip that section for now, other than mentioning the little bottom part of it. But what happens here is that initially it functions as individual bacteria where it uses flagella to move through the water or mucus to make contact with cells. It can use pili and cell wall adhesins to adhere, and then that allows them to gain a foothold. And as they begin to replicate, eventually there's enough bacteria where they start developing biofilm formation. And as the biofilm forms, the bacteria loses the individual characteristics like flagella and pili and start functioning as a population. And that allows them to produce more glycocalyx or biofilm. It allows them to obtain nutrients from host cells. And eventually, as it mushrooms up, it develops water channels and such, and you get an intact biofilm. And eventually, when that biofilm becomes too crowded through quorum sensing, some of the Pseudomonas again start producing flagella, escape from the biofilm, function as individual bacteria, and can colonize a new location. But again, the details of that we'll take up in Unit 2 under quorum sensing. But this just shows you that part of that process is the biofilm formation. And that happens after the quorum sensing genes are activated at a certain density of bacteria, and that's what allows the bacteria to persist in the body rather than to initially colonize. And two good examples of biofilm producers you're familiar with are Streptococcus mutans and Streptococcus sabrinus. These are two bacteria that are implicated in initiating dental caries or cavities. And here's where sugar comes into cavities. The sugar doesn't cause a cavity. The sugar we're talking about is sucrose, table sugar, which is a disaccharide composed of a molecule of glucose and a molecule of fructose. Now, Streptococcus mutans, one of the microbiota in our mouth, can use an enzyme called dextransucrase that converts sucrose into very sticky polysaccharide called dextrin. And that's what actually forms the biofilm that eventually enables the bacterium to adhere to the enamel of the tooth, forming plaque, and thus resist flushing. And again, in Unit 2, or Unit 3 actually, when we get to bacterial pathogenicity, we'll be looking at that. And then as the bacteria continue to grow and form a biofilm, these two species of Streptococcus ferment glucose to get energy. So they take the table sugar, use that to make their glycocalyx and biofilm to adhere and form a population. But Streptococci get their energy strictly from fermentation. And they ferment the glucose from the sucrose. In fact, they convert the fructose to glucose, so there's more glucose to ferment. And of course, they're fermenting glucose in order to produce ATP. But the end product of fermentation of these bacteria is lactic acid. And so now the lactic acid, the waste product of fermentation of glucose, is released onto the surface of the tooth, and the acid begins to decalcify the enamel of the tooth, eat a hole in the tooth, until eventually the bacteria can get down to the living part of the tooth, causing abscesses to form. So we see a couple of electron micrographs here. This is very early in biofilm formation where the Streptococci are adhering to the enamel of the tooth. Here's the chains of strep. And right now this glycocalyx is so thin you can't see it, but it's already allowing the bacteria to adhere to the enamel. Now, once those quorum sensing genes get turned on and the bacteria begin to replicate and communicate with one another and act as a population, they all begin secreting a common glycocalyx or adhesive matrix, and that eventually traps other bacteria. And eventually, when you have full-blown plaque, it looks like the electron micrograph on the right, where you see all kinds of different bacteria living as a population on the surface of the tooth. And of course, we spend a lot of money trying to get rid of that biofilm on our teeth, from dental cleansings to flossing to brushing to water picks and such. And so that's the link between sugar and cavities. It as the building block for the biofilm of the bacteria. and it becomes the energy source for the bacteria. And actually, it's the waste product of fermentation, the lactic acid, that causes the cavity. So the more sugars in the mouth, the more they're fermenting glucose, the more acid they're producing, the more they're eating holes in the enamel of the tooth. Here's another nice picture of a biofilm. This is Staphylococcus aureus forming a biofilm on an indwelling catheter. So we're looking inside the catheter tube, and here we see the clusters of staphylococci, but here's the biofilm they're secreting. So they're adhering to the inner wall of the catheter, and that allows them to colonize and persist, and then eventually spread into the body through the catheter. And there are many chronic and difficult to treat infections we think are caused by bacteria and biofilms. First of all, in the biofilm, the bacteria grow more slowly. They exhibit different gene expression than the free bacteria that started colonizing the body. And they're more resistant to antimicrobial agents because the antibiotics have reduced ability to penetrate the dense biofilm. And these have been implicated in a good number of infections, from tuberculosis to staphylococcus infections, Legionnaire's disease, and of course, periodontal diseases. And it's estimated that as many as 10 million people a year in the United States may develop biofilm-associated infections as a result of invasive medical procedures and surgical implants. Unfortunately, what often happens during surgery is that microbiota from the skin can get into the invasive medical procedure or get on the surgical implant, or they can get in there through glove punctures. And then they start forming a biofilm on the implant, and it's only later on as they begin to replicate and become populations growing on there that you see the results of the post-surgical infection. And there's a little movie there you can watch from YouTube on bacteria forming biofilms if you'd like to do that. And a little self-check you can do on biofilms. So that is our SoftChalk lesson on the Bacterial Glycocalyx, which includes capsules and eventually their formation of biofilms. And here's a self-quiz covering the whole SoftChalk lesson that you can do.